Recent years, mRNA vaccines have revolutionized our approach to infectious diseases, most notably with the 𝗖𝗢𝗘𝗏 pandemic. But what if these same vaccines could also help fight cancer? New research reveals that SARS-CoV-2 mRNA vaccines, originally designed to protect against 𝗖𝗢𝗘, can actually sensitize tumors to immune checkpoint inhibitors, powerful drugs used in cancer immunotherapy. This unexpected connection opens up exciting possibilities for improving cancer treatment outcomes, especially for patients whose tumors previously resisted immunotherapy. Let's explore how this breakthrough was discovered and what it could mean for the future of cancer care. Immune checkpoint inhibitors, or ICIs, have transformed cancer therapy by helping the immune system recognize and attack cancer cells. However, not all patients benefit from these treatments. Many tumors create an immunosuppressive environment that prevents immune cells from mounting an effective response. This resistance is especially common in so-called cold tumors, which lack pre-existing immune activity. Researchers have been searching for ways to overcome this barrier and make immunotherapy work for more patients. The answer, it turns out, might come from an unexpected source: mRNA vaccines. RNA vaccines, like those developed for 𝗖𝗢𝗘𝗏, work by delivering genetic instructions that teach our cells to produce a harmless piece of the virus, triggering an immune response. This technology not only protects against infection but also stimulates the body's innate and adaptive immune systems. Scientists wondered, could this immune activation be harnessed to help the body recognize and attack cancer cells, especially when combined with ICI? The research team set out to test this idea in both animal models and human studies. Investigate, researchers administered SARS-CoV-2 mRNA vaccines to mice with established tumors, alongside immune checkpoint inhibitors. The results were striking. Tumors that previously resisted ICIs became sensitive, showing reduced growth and, in some cases, regression. The key was a surge in type I interferon, a powerful antiviral cytokine which primed immune cells to recognize and attack tumor antigen. This combination therapy outperformed either treatment alone, suggesting a synergistic effect that could be translated to human patients. Type I interferon plays a central role in the body's defense against viruses and tumors. In this study, the mRNA vaccine triggered a strong interferon response, activating antigen-presenting cells and priming CD8+ T cells to recognize tumor-associated antigens. Blocking interferon signaling eliminated the anti-tumor effect, confirming its importance. Interestingly, the effect was not specific to the spike protein of SARS-CoV-2. The innate immune sensing of the mRNA itself was the main driver. This finding suggests that mRNA vaccines, regardless of their target, can act as powerful immune modulators. Tumor microenvironment is often hostile to immune cells, filled with suppressive signals that block effective anti-cancer responses. The mRNA vaccine-induced interferon surge reprogrammed this environment, activating myeloid cells and enhancing the presentation of tumor antigens. This led to the recruitment and activation of tumor-specific T cells, which infiltrated the tumor and initiated an immune attack. The result was a dramatic shift from an immunologically cold to a hot tumor, making it much more responsive to immunotherapy. Do these findings hold true in people? To answer this, researchers analyzed data from cancer patients who received immune checkpoint inhibitors, comparing those who had also received a 𝗖𝗢𝗘𝗏 mRNA vaccine within 100 days of starting therapy to those who had not. The results were compelling. Patients who received the vaccine had significantly improved overall survival and progression-free survival, both in non-small cell lung cancer and metastatic melanoma. This benefit was consistent across different vaccine brands and dosing schedules. Understand the immune changes in humans. The team collected blood samples from healthy volunteers before and after 𝗖𝗢𝗘 mRNA vaccination. They observed a rapid and robust increase in interferon alpha and other inflammatory cytokines, peaking at 24 hours post-vaccination. This was accompanied by activation of myeloid cells, dendritic cells, natural killer cells, and T cells. These changes mirrored the immune activation seen in animal models and provided a biological explanation for the improved cancer outcomes observed in vaccinated patients. One of the key findings was that mRNA vaccination increased PD-L1 expression on tumor cells. PD-L1 is a protein that tumors use to evade immune attack, but it also serves as a target for checkpoint inhibitors. By boosting PD-L1 expression, the vaccine made tumors more susceptible to ICI therapy. In large patient cohorts, those who received a 𝗖𝗢𝗘 mRNA vaccine before tumor biopsy had higher PD-L1 scores, making them eligible for single-agent immunotherapy and potentially improving their treatment options. Importantly, the effect of mRNA vaccination was not limited to lung cancer or melanoma. When researchers looked at a wide range of tumor types, they found that mRNA vaccines increased PD-L1 expression and improved survival in patients receiving ICIs across diverse cancers. This suggests that the immune-modulating effects of mRNA vaccines could be harnessed broadly, offering hope to many patients with different forms of cancer. Particularly exciting finding was the ability of mRNA vaccines to restore immune sensitivity in cold tumors, those with low baseline PD-L1 expression and poor response to immunotherapy. Patients with these tumors who received a 𝗖𝗢𝗘 mRNA vaccine before starting ICIs achieved survival rates similar to those with more immune-active tumors. This means that a simple, widely available vaccine could help overcome one of the biggest challenges in cancer immunotherapy. Makes mRNA vaccines such powerful immune modulators? It's not just the antigen they encode, but the way the mRNA and its delivery system stimulate innate immune sensors. The lipid nanoparticles used to deliver mRNA form complex structures that activate pathways like MDA5, leading to a cascade of interferon production and immune cell activation. This systemic immune reset primes the body to respond more vigorously to cancer and enhances the effectiveness of checkpoint blockade. Discovery that off-the-shelf mRNA vaccines can sensitize tumors to immunotherapy has major clinical implications. Unlike personalized cancer vaccines, which require complex and time-consuming manufacturing, 𝗖𝗢𝗘𝗏 marinae vaccines are widely available and easy to administer. This approach could provide a low-cost, rapid way to boost immunotherapy responses in patients awaiting personalized treatments or in settings where such therapies are not accessible. It also opens the door to developing new universal mRNA therapeutics designed specifically to reset the immune system for cancer therapy. While these findings are promising, there are still questions to be answered. Not all patients may respond the same way, and the optimal timing and dosing of mRNA vaccines in relation to immunotherapy need further study. Researchers are also exploring whether modifying the mRNA or its delivery system could enhance the immune response even more. Ongoing clinical trials and laboratory studies will help refine these strategies and determine how best to integrate mRNA vaccines into cancer care. Summary: the discovery that SARS-CoV-2 mRNA vaccines can sensitize tumors to immune checkpoint blockade marks a significant advance in cancer immunotherapy. Harnessing the immune-activating power of mRNA technology, we may be able to help more patients benefit from life-saving treatments. As research continues, this approach could transform the landscape of cancer care, making immunotherapy more effective and accessible than ever before. The future of cancer treatment may be shaped not only by targeted drugs, but also by the vaccines we use to protect ourselves from infectious diseases.